So this is going to be structured just like previous versions have been, which is I will do a little bit of review from the last time, kind of the things that are important that need to be reemphasized. I will also go into some new stuff about how we do short read alignment to a reference genome, and then talk a little bit about kind of the newer algorithms that are coming out now in areas where there could be improvement. If there is a bunch of time left at the end, I'll tell you a little bit about some work we're doing, which isn't exactly short read alignment, but is pretty conceptually related in a few ways that I'll mention. So if we make it there, that'll be great. If not, hopefully you learned something from the rest of this presentation. Okay, before I get started, does anyone have questions from last time? And I'll look at the chat too, since I think some of you are sharing questions there. Okay, here we go. Yes. Okay, you're talking about the research we did. The question is, you said that you evaluated the quality of genome assemblies according to several criteria. So those included misassemblies, misassembled contigs, mismatches, and others. And I compared them to a reference genome. And if there is no reference sample, is the same evaluation made in this case? How do you compare them? So that particular study was set up such that we knew the answer, right? The idea was to develop a new method for assembling genomes from very, very, very little input material. So very little input DNA and just one library from each of these things. So in that case, we set it up so that we would for sure know the answer, and that comparison, to the melanogaster reference genome should be completely reasonable, and it seems like it was. But that's a great question. How do I do this if I don't know the answer in the first place, right? Like de novo genome sequencing, the point is to discover a genome that you don't know what it is in advance. So there's a couple ways people do this. Typically what you would do is evaluate the completeness of your genome using something called BUSCO, which I don't remember exactly what that stands for, but part of that is single copy orthologs. So what this is a method for taking a genome using gene prediction models, which I also won't have time to talk about in these 3 lectures, but they exist. You can predict genes just from gene sequences. Once you predict those genes, what you can do is figure out, do I see all the genes I should? And what I mean by that is there's a few genes that are, sorry, my dog is barking in the background. Hopefully that's not too much of a problem. But there's a few genes that you can see that you will always see in almost any genome assembly. Those are called single copy orthologs. We always see them, there's always one copy. And so what you do to evaluate the completeness of your genome is you say, do I see all of those genes or most of those genes? And a typical BUSCO score can be anywhere from like 70% to 100%. Well, they never get to 100, 98%. Good modern genome assemblies tend to be in the range of 95% BUSCO scores. So that's kind of where we're shooting for most of the time. So that's another way that people do this. That and the other big metrics are the completeness of the genome. So for example, if I know that this species from its karyotype has 20 chromosomes, and I think my genome assembly is chromosome scale, then I should see 20 really, really big scaffolds that basically are 20 chromosomes. And if I don't see that, it suggests there's a problem. There's lots of reasons for that, but those are kind of the overarching ways that people go into evaluating a genome when we don't know the answer in advance. Great question. I really like that. Does anyone else have a question? And did that answer your question completely, Vlasenko, Galina? Yes, sounds like it did. Good. Okay. I will just get started. I think as before, I'll be better about watching the chat this time. So please just interrupt me with questions and I'll try to get to them as we're going through this. And hopefully that'll be good. Let's see, there's people in the waiting room, but I think someone else will probably handle that. Or I'll just admit them. Here we go. Okay, so today we're gonna talk about read alignment and a little bit about filtration. At the very end of this, what you should have is sort of the primary data object that you would use to do operations in a program called Angst or in other genotyping frameworks, but I think Torfin will probably talk about Angst. So the idea is to get you set up to go into that. Quick outline, there'll be some review. I'll just quickly go through some of the major things we mentioned to remind you how they work and why they're important for understanding genomes in the first place. We'll talk about read alignment, so kind of stating formally what the problem is, and then describing. I won't do a full explanation of the Burrows-Wheeler Transform today. We'll do kind of the short version of it. But if anybody's interested, there's lots and lots of good resources that have much more complete explanations that I can give you. So if you just send me an email after this, I'll be happy to find that information for you. At the end, we'll do some important challenges, sort of what the future of short read alignment is and why it's hard to do in some cases, sorting and filtering the output, and just a demonstration of how this works. So to review, I think I've emphasized this a lot, but it is super important, so we're going to do it again. FASTQ format, this is your raw data. It's what streams off the sequencer. Well, not exactly, but it's the thing you get back from the sequencer. And each line is defined with, or each read has 4 lines that define it. Sequence ID, the actual sequence, this spacer, this plus, and then whatever. And then finally a quality string in ASCII encoded values. And remember that these are Fred scaled qualities. So you can go from each of those, right? The A corresponds to the probability that this G is incorrect, the G to the probability that this A is incorrect, etc. You can use these, convert them to their Q score, and then use standard sort of mathematics right here to get that probability that site is in error. And that's probably the most useful thing for you that you'll want to know in genotyping, because you tend to integrate over all possible errors to figure out if we think a given genotype is correct. So really valuable for likelihood calculations that will come later. Okay. One other important piece of review for you guys today is that these very often, almost always, come as paired end reads. What that means is you'll have 2 files of reads. They're typically denoted with the same base file name _1.fastq and _2.fastq, or R1 and R2.fastq, something like that. That indicates that the first is the first in the pair, the second is the second in the pair. That order doesn't really matter. You can feed them into a alignment algorithm in reverse, give them the second then the first, and it should still do fine. But the important point is there are 2 files. They correspond to each other such that the first sequence ID in the first file should match the first sequence ID in the second file, and they have the exact same number of reads in both files. That is, every read has a pair. In some rare cases, some clusters fail on the second run, and so a read will have a pair, but its pair will just be a bunch of ends. So it will be no real sequence, it's just empty. And usually aligners just basically ignore it, but it still needs that pair there. So you should always have the exact same number of lines in file 1 as in file 2. Okay. One thing I did not mention that I really ought to have is FASTA format. So this is actually what comes out of your assembly program. And basically you can think of FASTA as the same as FASTQ, except that we don't represent the uncertainty in the sequence in any form, right? So what happens is there's a header line now, which begins with the caret, not with the @ symbol. And you have actual sequence that follows it. One other thing that really needs to be emphasized is, unlike FASTQ, which is really easy to read in because every read has 4 lines, you can just import 4 lines at a time and you've read a read. In FASTA, there's no such guarantee. There are as many cases where the sequence will stretch over many lines, and so you don't get the same kind of nice compact format in many cases. So you need to be a little bit more clever when you read in FASTA format in many cases. This is what basically all genome assemblies are in, and what that means is the implicit assumption we've made is the sequence is so high quality. We really, really believe our sequence assembly, to the point that it isn't important to understand any uncertainty in this genome assembly. So there may be sites that are wrong, but we're not going to have any indication that those are wrong relative to others. We're just going to move on with our lives and pretend the entire thing is completely reliable. Okay, so that's another major file format you will use. As I said, your reference genomes will typically be in FASTA format. When you make that assembly, what Velvet actually spits out is FASTA format as well. So you will use both of these. Okay. Quick review of overlap layout consensus. As I said, this was the first way people were doing genome assembly, and it's actually come back into popularity now, or versions of overlap overlap layout consensus have become popular again. And they've become popular for what's called long read alignment. And I'll talk about that in a second, or long read assembly. And the reason is you often have much lower depth of long reads, but often much bigger chunks that overlap between them. And the basic steps of this are exactly as the name would imply, overlap, layout, and consensus. The idea is you start by finding all possible overlaps. In other words, you do an exhaustive pairwise comparison between all of your reads. You then lay it out such that the graph is defined that each of the sequences is a node and the edges are the overlaps between them. And then finally, what you're going to try to do is take what's called the Hamiltonian path, where you want to visit each node once as you traverse the graph. That's not always possible, right? For the reasons we described for De Bruijn graphs, there may be repeats or other things that cause my graph to go in a circle back around to itself. When that happens, most assemblers split the graph if they can't resolve that in some useful way. They split at that site and your graph just sort of ends on some repeat element. Okay, at the very end, you take a consensus. What that basically means is I'm going to look at all of the reads that stack up at a given position in my consensus, and I'll take the average of them. So for example, you have 3 reads here. If these had A, and T at a given position, you'd probably take the A because you saw A twice and T once, and you say, well, A seems more likely. Okay. But as I said, this was not appealing to us when we started getting into next generation sequencing, and there's a couple reasons. One is your reads are really, really short, but there's also an absolute ton of them. So when we got into Illumina sequencing data, it has become extremely difficult to get 400, 500 million reads from $1,000 worth of sequencing. So you get a lot of data really fast, which imposes a totally different challenge. And it actually goes all the way back to the first step, which is that if you try to find the exhaustive pairwise overlaps between all possible Illumina reads in a run, that's a lot, and it will be extremely computationally expensive to do that. So that fell out of favor. We gave up on that plan. And we switched for a long time, and this is still extremely popular. So De Bruijn graphs are still used in genome assembly. I've used them in genome assembly. It is going to be less and less common as long reads really take over, but still very important. They really solved that key important problem I was describing, though, which is the overlapping problem. Because now what I do is I take a read and I say, here are the constitutive smaller sections. They're called k-mers, and I'll go back through that again, how we do that. But all I have to do to figure out if 2 reads might overlap is look and see if they contain the same k-mer. Right? If they have the same, say, 50 base pair sequence in them, there's a pretty good chance those reads actually overlap in the genome as well. And so what you've done is you've taken this problem of exhaustive pairwise alignment and turned it into a lookup table problem, which probably those of you with the CS background know is going to be a way, way faster proposition than the original. Okay, so it's nice. It's a much simpler way to do this. The other reason this works, specifically in the context of Illumina data, is that the error rate is typically fairly low. It's not 0, but you might get error rates in the range of 1 in 500 today to 1 in 1,000, depending on the machine, depending on the library, and lots of factors, maybe all the way up to 1 in 100. But even so, those are pretty low error rates, right? If I try to assemble a genome with a k-mer size of 31, then the error rate's 1 in 1,000, the vast majority of my k-mers will be exactly perfect, and the lookup table's ideal, right? I just have to say, does that sequence exist somewhere else? Okay. So to make a De Bruijn graph, what you would do is you would take your read and you would deconvolute it into the constitutive k-mers. In this case, we're showing you what a 3-mer, where k is a number, an integer number, and k must obviously, by definition, be smaller than the read length for this to work. And usually k is actually substantially larger than 3. K is normally in the range of, say, 31, 41, something like that. But anyway, the demonstration works with the 3-mer. So you find all your constitutive 3-mers, right? You have ATC, TCA, etc. And then what you're doing for each of these is you're constructing this little link in a graph, right? So your nodes now are your k-1 mers on the left and the right. And the edge is this k-mer. And it's actually a directed graph. We're going from GC to CG this way. Okay. So if we were to do that for 2 reads, and I'm skipping a step in here, but just imagine I've taken 2 reads. They overlap right at this CG in reality. and we throw them down on this graph, you can see that when we combine them, that is, they now begin to share nodes that are common between those two reads that have their own little de Bruijn graph. We combine these into one graph. You see that it has the same basic pattern. In fact, you can still basically traverse this graph in a single shot because you must start at the AT because nothing feeds back into it. AT, blah, blah, blah. Only way out is here. Now you have a choice. You can go to here, but again, it only goes that way. So if you want to visit all of these nodes and all these edges, you'd have to go that way. You'd have to go this way, up here, back around, over, down, and through. So you could solve this. And actually, the way people solve this in reality most of the time is to use the read overlaps, right? So there's sort of two different ways of traversing the graph shown here and here, right? You could actually do this a slightly different way, which is to go all the way forward and then back. Yes, 1, 2, 3, 4. Yeah, okay, so this traversed the graph slightly incorrectly relative to what we know the real answer is, which is over here. But the reads solve that, right? We see that this one overlaps the first bit, so we can be sure that that is one linear shot. This one overlaps the second bit, so we can be sure that it goes up here. So people often use reads or even read pairs to figure out which chunks of the graph go together after the fact by sort of aligning the reads back to their graph. Okay, just to review the key trade-offs here, usually the trade-off you're gonna make is that when you have a higher K value, it leads to higher specificity. And I think that's a fairly obvious and intuitive thought, but let's just try to explain it quickly. The probability of overlapping by chance with a much longer sequence, say 51 base pairs, is very, very small. But if your K-mer value is 3, there's a very good chance that two reads that have nothing to do with each other in reality share the same 3-mer. And the problem is when you try to make an assembly with a very, very small K value, you'll see lots of loops and lots of links in your graph. The other problem, the other side of this spectrum, is a very, very high K value where you have high specificity, but you may not even see overlaps between your reads, right? If your sequencing coverage is not that high, there's a good chance I won't get the right read that tells me there's a given overlap. So this is an example of that. Svetlana, hello. This is an example of that where these two reads only overlap these two base pairs. And so if you make make your de Bruijn graph with K equals 5, you will never see even a node, a K minus 1 mer that's shared between them, nor an edge that's shared between them. So it cannot be used. Of course, as I was saying on the other slide, the same thing applies in reverse. Imagine in the case shown here where we try to make our graph with K equals 2. You can see that this looks like a pretty silly graph, right? Where the nodes now are just A, T, C, and G, and there's a ton of links between everything. Obviously, this is not going to be a useful way to assemble a genome. So in practice, what we all do is just play with the K value setting in our assembler until we get something that we like the most. And in fact, there's a couple programs that you can look into, like IDBA, which is a de Bruijn graph assembler that actually runs a bunch of different K values and tries to combine those assemblies later to sort of get the best of both worlds. And it seems to work reasonably well. We've used it some and been very happy. Okay, cool. I alluded to this earlier, but just to mention it again, repeats are a problem, right? They'll actually be a problem for mapping, as confident mapping of short reads as well. But your basic issue is if you have a sequence arranged like this, where the two green parts are repeats, you're gonna have a loop in your graph, right? You'll come from the green back into the blue, back into the green, and then into the red. But it may not always be possible, depending on the structure of the genome and the reads, to resolve this in a single linear line that we're confident. It may very well be that we have to break the graph at this green chunk because we're just not sure how it enters and how it leaves. And you'll get what's called contigs, which will be the edges that we can be confident in. So yellow, red, and blue. And we'll have the green part broken up from those. Okay. Looks like there's a question. I read that okay. The question is, I've read that a GC bond is more stable than an AT bond due to the additional hydrogen bond. Is that in fact used when assembling the genome or somewhere? So the GC content does come into play in some cases, but the actual biophysics of how the bonding works is typically ignored in bioinformatics. But just for those of you that are wondering what we're talking about, GC are each other's pair, AT are each other's pair. GC has 3 hydrogen bonds, AT has 2. What this typically means is that regions of the genome with higher GC content are more stable, at least thermodynamically, and probably in other things as well, and AT less stable. And so you see this, for example, when you do PCR, there's enormous bias toward higher GC content sequences, presumably because they're better at binding to the primers and actually having the polymerase go across them. And so it can be really important for things like genome sequencing, where low, extremely high AT regions don't sequence very well. And of course, if you go to 100% GC, that doesn't sequence very well either. But it is not a fact we rely on heavily in genome assembly or short read alignment. That's a good question, though. I like that a lot. Cool. Did that answer your question, Galina? I'm going to assume so, but let me know if it didn't. Yeah. Okay. Okay, cool. So this is one of the statistics we use. I'm going to hammer it home because basically every genome assembly paper that you will read will report at the very beginning of their results section what N50 they got when they made their genome assembly. So your N50, to remind you, is the length of the largest contig, such that the length, if we sum the lengths of all contigs of that length or larger, it would contain half of the total assembly. Right, so this is the exact same example I used last time, but basically the idea is if we had all these contigs with lengths 2 through 10, their total length would be 54 base pairs. What you're basically trying to do is find the fewest number of contigs whose length in total sums to 27, right? So in this case, it's just 8, 9, and 10 is 27, so you're done. And 50 is 8. It's the very first statistic you will see. It's the first thing I would look at if I made a new genome assembly, is just how much stuff did I stick together. That's basically what this is trying to summarize with a simple, reasonably intuitive number. I'm not going to go into it in more detail than that, but just be aware it's a thing. Most assemblers will tell you the N50 when they finish making your assembly. The very rough rule of thumb is you'd like this number to be as high as possible. With that said, there are cases where that doesn't help you. For example, if your assembler is just greedily sticking everything together, even things that shouldn't go together, you may have made a lot of misassemblies in your genome, which may not be ideal. Certainly a problem. Okay. As I mentioned earlier, the optimal k-mer size is hard to know in advance. So usually the solution is just to run a bunch of k-mer values. This is this display, right? This is this N50 statistic. And in fact, the way I made this originally was I took a mitochondrion, a human mitochondrion, and some reads from it and just tried to assemble them with different k-mer values. And you can see that when I do it with 15, it's terrible. Clearly there was a ton of loops in that graph, could not be traversed. But when you do it out here with about 31, I believe this is, it does pretty good. In fact, that's basically the whole length of the human mitochondrial genome, so one shot. Okay. We're not going to go back and redo this, but just to remind you the basic steps we use. Actually, we will redo the first and second things in here, but the basic steps we use. You can download sequences from the SRA using FastQDump. Obviously, when you get sequences back from a sequencing center, they're not in a database yet, in which case you'll probably use wget, but you'll figure that out. As soon as you get your read data, the very first thing I think you should do, that I highly recommend, is to just run FastQC. Just get a summary of what the run looked like. Usually it tells you nothing because your run was fine. Every now and again, you will discover data where there's a real problem, and you need to be aware of that. And it's much better to figure that out early than it is to try to do a whole analysis and realize that it's just an artifact of some error in the library prep. So do it quickly. It's super easy to do. We showed you how to do it with Conda, and it's like 2 lines of code, so you can do this. Okay. After that, assembling a genome with Velvet. Remember, there were 2 main steps. The first creates your hash, or essentially your lookup table for all your reads. And the basic format of that command would be here. And the second is this Velvet G, which is the thing that actually makes the full graph and tries to resolve it into contigs. At the very end, if you take your assembly log and you grep out N50, you basically see how you did. It'll be in that log file. Okay, cool. Final piece of review, draft assemblies versus complete assemblies. So anything made between about 2009 and 2016 is basically a draft assembly. That's not always true, but the vast majority of genome assemblies were. And the reason is the reads were short and we didn't have good linking technologies. So what people ended up doing was basically stopping here at this step, which is the contig step. Now what's going on is if you have a long read that spans potentially a repetitive element between these two contigs, you can resolve that and get all the way to scaffolds. Or alternatively, you may have linking libraries to figure out how these things lay out. Even if I don't know the sequence between them, I can figure out how they lay out along a chromosome. But regardless, what you'd like to do, and it's becoming increasingly common today, is to get to a almost complete chromosome scale assembly. So you'll see those two terms a lot, draft assembly. All you need to know is that means it's probably not a great assembly and that you should be very, very careful when you're using that kind of data. Okay, cool, new stuff. So today we're gonna do an example of short read alignment. And the big problem here is actually in some ways simpler than de novo assembly. And I do it in this order because you have to have a genome to put your reads on. So it feels more natural to do genome assembly first. But I think short read alignment can be a little bit easier. And what the basic idea is that you already have a reference genome. So you've made a sequence from some representative individual in your species, and you've done a really good job, or you think you've done a really, really good job such that you don't have to worry about the completeness or accuracy of this reference genome. So for humans, we actually have a ton now. The first one was Craig Ventnor's genome and a few other people. But now there are many, many, many what we would call platinum complete genomes available for humans. Most other species have at most one, although that's changing as well. So be aware that this, you may have a choice in your reference. For human stuff, I recommend you just use the reference we've all accepted, which is GRCH. Is it 39 now? Look it up, NCBI has it. But the short version is we have a sequence. We know this sequence. Now, what we want to do is we want to take all these really, really short sequences that we make from a genome, right? We can't sequence the whole thing end to end. If we could do that, it would be really easy to do this sort of thing. We could just literally align the full chromosome to the reference. We can't. We actually sequence much smaller sections. And typically with Illumina sequencing, you'd be doing about 150 base pairs at a time. This is an example, of course, so 6 base pairs is probably unrealistic. You are very unlikely to find unique alignments of those, but it's much harder to display larger ones and show you how this works. So bear with me on that. But what you want to do when you're doing this is take these really short sections. They're incredibly short relative to the reference genome, and efficiently search the whole reference genome to figure out where this read might go and where the best possible alignment is. And that is actually a challenge. So you could do this with like hashing the entire reference genome and just looking up where things are. And that would totally work as long as your reads were perfectly accurate. But they're not. They have some rate of errors. And I would add that that's probably still not the most efficient way to do this, although I think it can be pretty efficient in some cases. In fact, the first aligners were based on this. So MAC and I think the original SOAP model was both hash-based. They just made a hash of the genome, looked up where reads might go, and found the best alignment around there. So what we're showing you here is what we're looking for with this sort of thing, right? So if you go out here, you see a case where there is a SNP relative to the reference. That is, every single read in my sample has a G, but the reference has a T. So we can be pretty darn confident that this is at least a heterozygous, if not a homozygous, difference relative to the reference genome. In contrast, we've got a T here. That T is probably an error. That's actually going to be a problem for Torfin, is telling you how to figure out where we think confident variants are and how to analyze them. But the important point here is that we have to, for this problem, we have to very efficiently figure out how these things go down in the genome. Okay, so those are the basic requirements. So we have to efficiently find all the possible locations for a sequence. So it's not good enough to find one, although many aligners only report one. They still need to know that other possible alignments were possible. And the reason that they have to do that is that you need to be able to represent the uncertainty for these alignments. There may be several positions that are equally likely. You want to figure out which of those positions you want to be able to quantify your uncertainty and tell me something about how sure I am that that read goes there. And this actually comes back to the thing I think I've said probably too much in this class, which is that repetitive elements and sequences that occur many times in a genome are your big challenge here. Right? If you sequence something that comes from the middle of one of those repeats, there's a very good chance that you cannot be confident in its placement, and it actually has many, many, many possible placements across the genome. You want to know that so you don't just stare at errors later. Okay. So how do we do this? A lot of, at least for Illumina reads, a lot of the approaches are based on what's called a Burrows-Wheeler transform. So the way that a Burrows-Wheeler transform works is you take some text, right? The example here is the word banana. And banana, so probably most of you know this, I'll just say it for completeness. When we append this $sign here, it means end of string, right? It's just to keep track of where this string has ended. So to make a Burrows-Wheeler transform, you actually take this, this string, this banana plus a $sign, and you find all possible rotations of the string, right? So we've, I guess this would be anana, $sign B, nana, $sign ba, etc., right? And so the idea is we see all possible ways of arranging this. And what you're going to do then is you're going to sort this sequence, right? And so you're sorting it by this first character, right? So the $sign always comes first, A, B, N, right? So that would be alpha in Latin, this is alphanumeric sorting. A comes before B, comes before N. Hopefully you all know that, but if you don't, you know it now. This is now sorted alphanumerically. Okay. Once you do an alphanumeric sort like that, this has some really valuable features, right? So if we look at the end of this, we have this, the Burrows-Wheeler transform of the string banana is A, N, N, B, $sign A. The reason this matters is that what it's done is that it has put characters near each other that are the same character, right? So in this sort of toy example, there's two N's and two A's. And the reason that that happens is because when you left sort, right, you sort on this base, that often tells you what character comes next, right? So in the word banana, N is followed by A in many cases, A is followed by N in many cases. So when you do that sort, you put your N's together, you put your, you put your A's together, and it becomes really easy to encode this in a very compressed format. So the way this compression works is you can do it with just run length encoding, which tells me basically that, okay, there's two N's. So rather than storing N twice, I can store N and then the number 2. And I know that it extended for 2 bases. This is a toy example, right? So there's only 2 N's in a row and 2 A's in a row here. But actually, when you do this on a whole genome at scale, you often put a lot of characters that are the same in a row. And there's 2 reasons for that. One is that your genome is only made up of 4 characters, A's, C's, G's, and T's. So there's a very good chance you're going to see stuff go together like you'd like. But also it's much, much larger. So when you do this full sort, you'll often see thousands of A's in a row that can all be recorded as A 1,000 times over, which is very, very convenient. And it's actually just a tidbit for you all. This is basically what BZIP is doing. That's what the block-based compression of BZIP is. Or BZIP 2 now. I don't think BZIP's still a thing. Okay, so so that actually is to say that the Burrows-Wheeler transform was actually a way of doing lossless compression. That is, you could compress this string, and I won't go into this today, I won't have time, but through an approach called LF mapping, you can go backwards. You can take that string and figure out what the original sequence was just based on this compressed string, which is really kind of amazing when you think about it. It's a little bit complicated, but not that bad. Highly recommend looking into it because it's sort of interesting. Okay. It's worth pointing out that this doesn't always work. So if you take this word, I've actually never heard the word before, but it's an example people often use, appellee. If you do this and you make your Burrows-Wheeler transform, so again, we append the $sign on the end here, and then we make all possible rotations of this. And now we're going to sort all possible rotations and put them out here, A, E, 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 L, L, P, P. Interestingly, this side has a bunch of nice sequence runs, the E's, the L's, and the P's. But the Burrows-Wheeler transform is over here. And in that case, you're not so lucky. You don't see that at all. You see E, $E, L, P, blah, blah, blah. There's really nothing there for compressibility. So it will not work in this case. But it often does. It usually works very well on genomes. And it's something we like to use for this reason. Okay. Probably many of you are familiar with something called a suffix array. So a suffix array is a Burrows-Wheeler transform, basically. The only difference here is that rather than just displaying the suffix, which was the end, you display the full rotation. But you can see that these two things clearly map onto each other almost perfectly. So a lot of people actually use the terms nearly interchangeably, which is why I like to mention this. Sometimes you'll see suffix array, sometimes you'll see Burrows-Wheeler transform. It doesn't really matter. Okay, so like I said, we're not going to go through the whole thing today. There just won't be time, but I do want to tell you kind of how, at least in broad strokes, how this can be used to efficiently search a genome for the possible placement of a read. And the way this basically works is, here's my sorted suffixes down here now for this sequence here, GATG, blah, blah, blah. When we make the Burrows-Wheeler transform, so in other words, what we're doing is we're displaying the base which precedes that suffix, right? So G is at the end, TG is the last two, so you see TG end, right? If we want to do this and we want to efficiently search the graph, something really nice has happened, which is that this is now sorted for us. So when you sort it, let's imagine that we have this simple case where we want to figure out if there's a sequence GA in our graph, or sorry, in our Burrows-Wheeler transform. This is not a graph. Burrows-Wheeler transform. You can actually just search the graph by bisection, right? Because we can actually just start at the middle and say, okay, is this string greater than, equal to, or less than this other string, GS? And in this case, you say, oh, it's less than, so we need to go to the right. So we'll go halfway to the end and we'll search there. And we say, okay, well, it's actually greater than this. Okay, we come back and we find out that, okay, in the middle now, at this position 7, we see our string, GA. So we can be pretty confident that, in fact, GA does exist in this sequence. And in fact, it exists twice. And if you wanted to exhaustively search the graph, all you would have to do, now that you know that you're at GA, is search the two edges, right? So we go here and we say GATG. Well, that's not the same. We're done. But if you go to the left, you see GA again. We can be pretty darn sure that it actually occurs twice in this sequence, which is pretty cool. So this is now an it's a log n complexity problem. And so you can get through this very, very quickly relative to sort of a naive version where you just read across the sequence and say, do I see a GA at this site? No. Well, what about the next one? No, etc., which takes forever. Okay. You were probably asking yourself the question, well, how does that actually solve the problem? You know, earlier on, I said when there are these hash-based versions of this where you have to find exact matches, it doesn't work. And you're totally right. So an exact match is not the way to solve this problem, because the whole point of this exercise is discover variation relative to a reference genome. So if all I did was look for exact matches, I would never find anything interesting. So what you need for all of these approaches, and almost every short read aligner has some version of this, even the hash-based ones, they do what's called seed and extend. So what they're looking for is an exact match of some length, right? So in the example we're showing you here, it's 3. This is often called a super maximal exact match, or SMEM. You'll see it written that way. But basically what BWA and many other programs are doing is they're starting with ACG, the exact match, and they're figuring out where that goes in the genome. Do we see ACG? Okay, we do here. Now we're going to try to extend the alignment, and it actually, BWA uses blast-like rules. There's a few ways you can do this, but usually what it is you have some penalty matrix and you do a Burrows-Wheeler alignment to the edges. The reason this is so valuable is that rather than doing a comprehensive Burrows-Wheeler alignment of my short read sequence against the whole genome, I've prioritized a few spots where it might really go, because I found one of these super maximal exact matches of some length, and I can just optimize that alignment locally in a relatively small part of the genome very quickly, rather than if I tried to align this thing to the entire genome perfectly from the get-go, it would take forever, and it would be a very hard problem. Okay, that's the short version. If anybody wants to know more about Burrows-Wheeler transform or BWA or how they actually work, you should send me an email. I'll find some really good references for you. There's tons of stuff out there. For today, we're going to move on and just show you kind of what you'd get out and how to do it. So you will have what's called SAM file format after you make an alignment of short reads. There are new formats that people are using, but this is sort of the most common de facto standard of the field. This is what angst reads in many cases, or at least the binary version, and it's very common. You should know how it works. SAM is the human readable version. So we don't actually store SAMs very often. We normally store what's called a BAM format file. The idea is to tell us something about our alignment and define that alignment relative to the reference genome. So you'll have a few different columns here. You'll have your header rows up here, and your headers typically define your reference sequence, the length of each reference. So in this case, there's only one reference contig. It's called ref and has a length of 45. It's telling us if this file has a sort order, so you could sort by read pair, you could sort by coordinate. This one is sorted by the coordinate. In other words, it's the read position on the reference genome. Okay, so those are your header lines. They all begin with this @ symbol. The actual alignments are below that. This block here, it defines our real alignments. And so you'll see things like the Q name, that's that sequence ID from your FASTQ file. You'll see the chromosome, R name, the reference name. This is the chromosome, the third column, that the read is mapped to. This is the position. So this would be the one base position in the SAM file, the leftmost position that the read maps to. This is your mapping quality. And I'll spend a second on that and just say, your mapping quality is defined as the probability that a given alignment is correct. And then it's in Fred scaled quality. So if you see 30, it means the probability that this is incorrect is about 1 in 1,000. Or in other words, we really, really believe that alignment, right? If it was 20, it would be 1 in 100. If it was 10, it would be 1 in 10. Okay. After that, you see what's called a cigar string. And cigar strings are a little bit horrible, but basically what they do is they define the nature of my alignment, right? So there's 8 matches, 2 insertions, 4 matches, 1 deletion, 3 matches. So it tells me how this read sits down on that reference genome. And they all have various different forms of that. Here we see 5 soft, 6 match. What that means is that only the last 6 base pairs here have been aligned to the reference. These 5 are what's called soft clipped, which means that you ignore them for the purposes of the alignment. The other way that can work is what's called hard clipping. So down here, we've actually just removed 6 base pairs from this read, and there's 5 matches here. Okay. Those are the main things you need to know about. There are others that go into these files, but the most fundamental things are where the reference, what reference contig we're on, what the position on that reference contig is, the mapping quality on that contig, the cigar string. That's what you need to know about for now. But again, this just defines the alignment. Each read can actually have more than one alignment in a BAM file. The difference, usually what will happen is if you have more than one, other alignments are marked as secondary. So your genotyper will know to ignore this for genotyping because we know this isn't the primary, the highest quality alignment in the sequence. Okay. A little bit more on cigar strings. They can be really ugly and complex. Not always. Ideally, with Illumina data and a reasonably closely related sample and reference, it looks pretty nice. One thing I like to emphasize is that, well, we'll go through this and then I'll emphasize it. You've got a number, then a letter. Basically, it's run length encoding as well, where an M would indicate a match. That is, there's a 1-to-1 base alignment between the read and the reference. The word matches can be a little misleading here. It does not mean they have the same sequence. All it means is that the alignment is 1-to-1, but there could be a difference within an alignment block between the reference and the read. There's just no character for that because it doesn't define an alignment, it defines a property of the alignment. Okay. The other characters are a little bit more self-explanatory. D would be a deletion in the read. I would be an insertion in the read. H indicates hard clipping, right? So in other words, we've removed part of the read altogether. And S is soft clipping. The vast majority of the time, you will never see hard clipping. Most aligners just do what's called soft clipping. And they do that at the ends of a read where maybe an error makes it so that it's very hard to see exactly how that should align, and we just throw it out. There's other reasons as well. Okay. So again, cigar string is the thing you care about here. It defines the nature of your alignment. I'm now giving you the graphical version of what I showed you earlier. So this is the exact same BAM file here at the bottom. Excuse me. Same BAM file here at the bottom, but in this case, what we've done is we've actually drawn out how those reads are aligned onto this reference genome. So to go through this really quickly so you understand what we're talking about here, this first one, read 001, right? It's on the ref chromosome, so that's this chromosome. And the leftmost alignment on that is base 7. So you see it starts here at 7 in one base encoding. Okay. From there, you can see that you've got 8 match, right? Your cigar string is here, 8 match. These 8 base pairs are aligned all the way to this 2 A's. Then there's 2 insertions. So what's actually happened here is this A gene in the read does not appear in the reference, and we've inserted that. Okay. If you go back down, you see there's 4 match. So if we follow along this string, it was 8 match, 2 insertion, 4 match. 4 match here, 1 deletion, right? So 4 match, 1 deletion, and then finally 3 match at the end. That's a cigar string in a nutshell. It's basically how you can do this thing, how you can define an alignment with just a string that tells me how this read sits on the reference genome when I made my alignment file. What you'd like to see are things more like what's at the bottom here, 9 match, right? That's really ideal. That 9 match, what it means is basically that that entire sequence has an exact 1-to-1 alignment across its length. It's very interpretable how that alignment should work. So usually that's what you're looking for, and it would be best. Okay, one other really important step that we will do that I want to just emphasize, often the way a library is made includes substantial amplification. What that means is you ligate your adapters on the edge, but then you do PCR. And if you're unlucky, one of those molecules will do really well during PCR and will be copied many, many times over, where other molecules around it are not. When you align that to a reference genome, what that should look like is reads whose first and last coordinate, or if they're paired, first coordinate and first coordinate of the second read, are exactly the same. The reason is you've copied that same original molecule. And so when you align it back to the genome, it should have the same alignment. And so a really important post-processing step, after we've made our whole alignment, we have our first BAM file, is to go through that BAM file and identify reads where the read and its pair have the exact same mapping coordinates. This suggests that they are a PCR duplicate and should be removed. And so there's many, many ways to do this. This is from the GATK guide, but of course there's Picard tools is how most people do this. There's actually a module I'll show you today within SAM tools that does remove duplicate step as well. But basically you want to go through your BAM file and throw out all the duplicates. What is the difference between clipping and deleting? So Dimitri, that's a very good question. The answer is, if we go back, hard and soft clipping, there's no term for deleting, right? Deletion is defining a type of alignment, right? So deletion would be, for example, this read doesn't have a base that aligns 1-to-1 with this reference genome here, right? So it's defining when something is missing in the read relative to the reference, we'd call it a deletion, right? So all of these strings are defined relative to the reference. So there's 2 insertion, that's 2 inserted in my read relative to the reference. 1 deletion is there's 1 deleted in my read, there's no base here relative to the reference genome. The clipping is actually the edges of the reads. Often we don't align the edges of the reads for a ton of different reasons. It's just much harder to be confident in alignments towards the edges of your reads because you don't have this context around the alignment, right? It just ends somewhere. So as you approach the ends, if you imagine that there may be an error in the last 5 base pairs, it can be very hard to know how that last part of the read should sit down on the reference genome. And in fact, it's typically removed by aligners. And so what clipping is doing is it's going through and removing that from the alignment. So for example, here, 5 hard is a type of clipping. It's just removed those base pairs altogether from the read. I find it mildly confusing that there is both hard and soft clipping, but there are. So so you got to work with it. But yeah, but the challenges are that they're showing you in the alignment here what those bases were. right? This is the 5 hard that was removed from this read down here, RAF03. And what you can basically see is that they don't actually match the reference at all, right? G, C, C, T, A. That's not what's sitting there at all in my reference sequence. And so the aligners decided to just remove that from the alignment. Does that make sense? That's the big difference here. Let me know if it doesn't. I see that. Okay, thank you, Dimitri. Cool. Okay, so to recap briefly the main alignment steps before we give you a short exercise and then do some alignment steps. These are 1, index the genome. So we're gonna create that Burrows-Wheeler Transform index and actually a bunch of other files as well. What they're really formally making is what's called an occurrence table, which is sort of a very, very short way of storing the information in that index, and a bunch of other stuff. Okay. Then what you're going to do is align the short reads. So you take your short reads, you align them to this index of the reference genome, you look up all the possible mapping positions, and you extend the alignments. Okay. We will then sort the reads by position, right? So when a genome gets sequenced, it's not like the sequencer knows how to organize the reads based on where they came from in the genome. It's completely random across this whole glass slide that it's sequenced on. And so they get sorted by their position on the reference so that reads that come from nearby places on the reference are nearby each other in this file. It's very convenient for all sorts of things that we'll show you. You then remove the apparent PCR duplications and potentially filter by mapping quality, but often the tools that you will use downstream will do that implicitly or explicitly as well. So you may not need to do that, but I'll show you how to do it today anyway. Okay. And just to remind you what the basic formats of these things will be, your indexed genome will be in FASTA format, your reads will be in FASTQ format, your index will be a type of Burrows-Wheeler Transform plus a bunch of other stuff. Those 2 things will go together to make a BAM file. When you sort the reads, it's still a BAM file. If you remove duplicates, it's still a BAM file. So kind of everything downstream is a BAM file until you start analyzing the actual alignments themselves. Okay. So this is going to be a very short, small group problem. You've got the following genome sequence, AGA, CT, blah, 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 and you are aligning the following short read. Ignore the fact that there are 2 strands, right? So just consider everything in the same strand for now. What are the most likely alignments? I'll show you the first one just for simplicity. You don't need to do a full Burrows-Wheeler transform. You can basically just look at this and see that GAC-TGG, well, that occurs here. So that must be one possible alignment because there's an exact match. So I want you to write out the cigar string for each and tell me what is the mapping quality for each alignment. And so just to remind you, this is the probability that the selected alignment is wrong. And it's not just that probability, it's the Fred scaled probability that the selected alignment is wrong. So figure out how to do that and get back to me. And I'll just send you off now to random breakout rooms. Hopefully that will work for you guys. And I'll bring you all back in about 10 minutes. I think this is a pretty doable question, but if you have any questions, let me know and we'll figure it out. Thanks. Okay, it looks like everyone is back. I can call on people, but does anyone want to try this one? Hopefully it was pretty straightforward. No, okay. All right then. I'll give you a start here. So I did the first one already, right? And we figured out GAC-TGG is this. You can also just sort of quickly scan across the sequence and see that there's actually one other occurrence of that, GAC-TGG right here. I would suggest to you that those are the only 2 even pretty close sequences in this entire genome. So probably those are the 2 alignments we're going to work with here. So I would say those are the 2 most likely alignments. So what is my cigar string? Right, remember the cigar string just defines the alignment. It doesn't tell me where it is. That's another part of the SAM file format. So in this case, the cigar string, if you say GAC-TGG, you see that there's a 1-to-1 correspondence across the entire sequence. So your cigar string would just be 6M or 6 match. And the same is actually true here. GAC-TGG. So you'd get 6 match again. Okay. So if we say those are the only 2 alignments we're going to consider, and I think that's probably pretty reasonable given the structure of this genome, what would be the mapping quality for each alignment? And the secret to answering this question was to realize that we don't represent any of the uncertainty in this reference sequence. So the fact that, so if there is any uncertainty here in our read, I didn't even show you a quality string, but it doesn't matter. You don't need that information because you'll see that, okay, it perfectly matches here and it perfectly matches here. In that case, it seems like the probability that one alignment is incorrect would be the fact that we can't choose between 2 alignments with any amount of certainty. Right? There's 2 that are equally good alignments. So the probability of an error would be 1/2. And usually the way that's represented, because of converting to integers and things, would be Fred 3. Right? Probability 1/2 turns out to be Fred 3. If you take log 10 of 1/2 times -10, you get about 3 when you take the integer. Is that kind of making sense? It's more or less what we're doing here. Okay. Okay. Cool. Thank you, Dimitri. All right. We're going to do an in-class example as well, which hopefully I've got sort of queued up. I need to send you all a file actually to do this. So we'll stop the share briefly. And I'm just looking at myself here. That's not great. Okay, in the chat, we will do send file. Give me 1 second. Okay, so we're going to do this alignment example. Okay, so everyone should have this. I certainly have it on my other Zoom window here, it looks like. It's the same sort of thing as last time, where it's a very short text file where I've put comments in to tell you why we're doing a given step. And then the actual command is not commented. So you could just run the entire thing using bash, name of the file, and it would run on, as long as you're on a bash shell. If not, you can figure it out. There's some other way to do this. Okay, and with that, I'll go back to sharing my screen and I will show you what we're doing, right? So, okay, that is, you can see my terminal, great. Okay, I think I'm even in the right place. Let's check. I've actually done this recently. Let's remove that. Okay. Cool. So this is our alignment example. This file you should now have on your computer, right? And so what we're doing here, I'll just read you through it really quickly and then we'll actually do it. We're installing the packages we need just like last time. If you're in the same Conda environment as last time, there's a good chance you already have SRA tools. You probably already have wget, but I'm just doing this to make sure we don't have any issues downstream. And then we're going to go through this. So what we're doing, you can see, is downloading a reference genome from this place, NCBI. But to start with, I'll just copy these commands. I'm going to do the Conda commands right now. So we'll do Conda installs. I'm giving it the channel explicitly with -C Anaconda. You may have these channels in your defaults. I don't know, but this should work for everyone, I believe. And it's nice just to see how to do Conda things in case you haven't. Okay. Tools E doesn't have this from last. What did I do? It looks like there must be a misspelling of some kind. Okay. Anyway. Sorry, I'm not as well set up for this as I ought to be. Desktop runs. Okay. Okay, so we're back here. We're going to start running these things. So the first thing we're going to do is get a reference genome. And this is actually the E. coli reference genome. So the most common bacterium that we study in general. Done. It's a pretty short genome. It's bacterial and it's gzipped, so it's real small. All right, if we hit ls-l, you'll see that it's right there. Pretty small genome. Let's do LH, so it's a little more readable. 1.4 megabytes of space. Okay. I want to remind you all of something. So we're going to take a look at this genome really quickly. And I'm using zcat because it's a way to decompress a gzip file and pipe it to standard out. But I don't want to read the whole file. That will just be gigantic and fill up the whole screen. So we'll just take the head. We'll look at the first 10 lines of the file. And if we do that, you get something like this, which is the actual FASTA sequence here. And it's telling us again, as I said before, the header starts with this caret. In this case, it's this reference accession here. And I think for E. coli, there may only be one, but I'll show you how to check that in a second. And then it's the sequence, right? And I said that a FASTA sequence can extend over many lines. So we have some characteristic length of sequence here, and then it's broken up over many lines. I actually find that extremely annoying, but I know there's some good reason for it, so it's still there. If we do this, what you can do is see all the contigs in the file, right? So I've grepped out that header character, which should never appear in a sequence. Remember, a sequence can only really be A, C, T, or G, and sometimes N. And so if you grep on the caret, you should see all the contigs in your file. In this case, there's just one. So one chromosome, it's circular, it's bacterial, so it goes all the way around on itself, and that's it. What we also need to do is to download some read files, right? So the first thing we did was copy that, but let's also do this. I'll try to slow down a little. I know it's probably hard to follow me as I'm jumping back and forth between things. So you may remember this command from last time. It's fastqdump. gzip, that is, we're going to gzip the resulting output files. So --gzip here, this section. And then split files, right? So these are paired end files. I know that because I looked it up in advance. And so what this will do is it'll take each spot. So each pair of reads is represented as one spot in an SRA file. It will separate them into two read files at the end. And then I'm giving it an SRR accession. So SRR 129.04594 is one that I know is a relatively small E. coli file. Okay, so let's do that real quick. And if I hit that, it's actually going to take a second because it's a little bigger than some files we've dealt with, but it will finish relatively quickly, hopefully. Let's see. Somebody has entered the waiting room. Admit them, sure. Okay. So that's taking a longer time than usual. My internet connection must be getting unstable today. That's not ideal, but hopefully we'll get through this relatively quickly. And we'll get to the end here. For those of you that may not know this, you can also put commands into the background. If you hit Control-Z, you will pause it. If you hit BG, it'll begin running in the background like that. This can be really useful if you set up some command and didn't remember to start a screen session or to put it in the background from the beginning. You can easily just stop it with Control-Z and BG. And then it'll just run until it finishes while you do other things. So if we hit LS, you can even see that this is currently building these two files that we're looking for here. And they are up to, oof, they're getting big. Did I pick the wrong one? 34 megs. Yeah, okay, I picked a relatively large file, sorry. That's on me. There was a few I was looking at and I did this. But in the meantime, while that continues to download, let's work on the next step. So what we're gonna do, as I said earlier today, is we need to create these indexes, right? So your index files are made with the BWA command, BWA command. And so it should just be BWA index and the name of your reference file. And it's okay to give this a gzipped reference file. It knows how to read gzip and still create the appropriate index files from that. So if we do this, it'll go relatively quickly. It's constructing your Burrows-Wheeler transform for the PAC sequence. Okay, we're done. So that's the whole thing. You've made your indexes. So if we hit ls-ltra again, you can see that we're still downloading our read files in the background here. They're getting, okay, we just finished. They're getting a little bigger, but also we've made all these other files here, right? So what these are compressed versions of those indexes I was talking about. I said there's actually many, and that's because there's a few different indexes you need here. But the key one is this Burrows-Wheeler transform, which is then turned into occurrence tables and other things later on. But it needs all of them. And the way you refer to these collectively is with the base file name here. You can see that all it's done is create those index files, but then also appended a new name onto the end of it. So Burrows-Wheeler Transform is here, pack, etc. Other things I think are kind of interesting are these index files are a little bit larger than the reference genome itself is, right? So 4.5 megabytes, this is 1.4 megabytes, the entire reference genome. So it can be that it seems inefficient at some level. Okay, cool. So we finished downloading our read files. These are here. We're going to just take a look at them really quickly to make sure we understand them. Again, those of you wondering how I'm completing this string really quickly when I'm typing, I'm just hitting the tab character and it's auto-completing the string based on what's available to the shell to complete. So if I do that and I look at the first, let's say 8 lines, that should be the first 2 reads. And in fact, it is. And you can also see that they are, for some reason, 75 and 74 base pairs in length. And they have this name here, right? So it's this really convenient name where the first part is the SRR accession and then .1, so the first read pair in the file. The SRR accession, this thing we downloaded earlier, and .2, second read in the file. And if we look at the other file, right, the paired end file, we should see the same thing. And in fact, we do, right? They have the exact same names, .1 and .2. So the sequence IDs exactly match between the 2 files, which would make sense because, as I said earlier, every paired end read file must have exactly 2 reads that match at each line. Okay. Okay. Cool. So what we're going to do next now, we need to align these reads to this file. And so the way we can do that is with what's called the mem command, which is a slightly modified version of the original BWA align command, but has the same basic underlying principles. And then what we're going to do is we're going to give it the reference genome. And in fact, like a lot of bioinformatics packages, if I just call it like this, mem, it'll tell me how to run the program, right? So there's quite a few different things you can do. But the important thing to do is this thing here, your IDX base, that's your indexed reference file that we started with. In1.fastq is the first read file, in2.fastq is your second read file. As you see, this is optional, and the reason it's optional is because you may have single end reads. We don't, we have paired end reads, so that's what we're gonna work with. We're not gonna modify this otherwise, and in fact, almost none of these other modifications are ever used. Most people run it on default, but that's okay. Still good to know it's possible. So if we do this, what that will do is it will output to the screen to standard out a SAM file, right? So it's gonna just start aligning the reads and outputting a bunch. We don't actually want to do that for many reasons. The first is that's not useful. How will I capture that? But the second is it's a really big file. So let's do something more clever. And what we're going to do is take that and we'll just store it, right? So we're going to tell SAMtools view that it's getting a SAM file in and it should write out a BAM file. And we'll just give this name. Okay, so what I'm doing here is I'm doing that alignment command, but I'm taking standard out from that, which is a SAM file, and I'm piping that into the SAMtools view function, which will read in a SAM file and output a BAM file. It'll read the SAM from standard in if I give it this function, this option here, this dash, and it'll output to that BAM file. So if we do that, it will also, I'm just going to let it output the log to this screen. You might also care to capture the log file, which you can also do separately. So we start seeing some really useful information quite quickly. It'll align sequences in batches. So SAM or BWA will read in a certain number of bytes of characters of reads and align them all in batches. And at the beginning, what it's going to do is it's going to tell us how it mapped the reads, right? So forward would mean both my reads point the same way on the reference genome. Forward reverse would be that they point at each other in convergent orientation. Reverse forward is pointing away from each other, and then reverse reverse is both pointing back. What you care about is you want the vast majority of your reads to be forward reverse. The reason is because the way we construct these libraries, these sequencing libraries, my reads basically should point at each other. They should be the 2 indexes that I have ligated to this given section of DNA, and then I read in from the edges. If I take those sequences that I read in from the edges, right, the reverse one will kind of look like this. They should align pointing at each other relatively nearby in the genome. And one very nice thing for this library is we see that the vast majority of alignments are actually like that. There's a few that are different. There's 23 that are forward, but there's 39,000 that are forward reverse, which is what we would like to see. So it means we did pretty well, actually. Below that, what you'll see is that it's computing the insert size distribution for each of these. In other words, it's computing on average how far apart those two reads are. So if we go down to this section where it analyzes insert size distribution for forward reverse, what you can see is that they map like this. Relatively close by, they're on average 184 base pairs apart from each other, and with a standard deviation of 89 base pairs. It's actually a pretty variable library, but it did pretty good. Okay, and that's for that first batch. This will keep doing this for each batch of sequences that it aligns. You're just coming out to the screen here. You see more. Ideally, each of these would be about the same because your read should be more or less randomly distributed in your file with respect to quality or position and all sorts of things. And you see that that's pretty true, actually. Here we see about 38,000, 39,000 sequences of that batch were forward reverse, good. And we get almost the same insert size. It's actually a little smaller. I'm not 100% clear on why, but you see that it kind of settles into that pretty quickly. So 167, 167, 163, down here. Again, we've got kind of in the range of sizes we would expect. Okay, it's also finished while I was talking about it. So that was our log that I just spit to the screen, but you might store that if you wanted to. Let's look at the files we have now. So ls-ltrah, we can see that we've created a new file, right? It's a BAM file. So as I said, this is the binary version of that SAM file. So you can't actually read this. It's not human readable, but it is a really compact way to store this. The corresponding SAM file might be as big as about a gigabyte. And so it's almost always advantageous to just store the thing as a BAM file rather than a SAM file. All right, so as I said, we got to do a few more steps to be happy with our alignment file. And the first step we're going to do, we're actually going to do is called samtools sort. So samtools is a suite of functions for modifying and manipulating BAM file alignments. So what we want to do is samtools sort, and we don't know how to run it, so we'll just do this again. These are really good bioinformaticians, so that they make it very easy for me to figure out how to run the program by just not telling the program anything useful. And so that's how this worked. So the samtools sort function works like this. We see that we can do a few different things. For the purposes of this, all you need to do is give it the input BAM file. And then what we're actually going to do is capture that as well, right? So what I recommend is you just label them as you go with what you've done to the file. So in this case, it has the exact same name, except I said .sorted.bam. There's a bunch of different ways you can do this. It's up to you, but I find that to be one of the easier ones to record what I'm doing as I go. And hopefully this will go relatively quickly. So as I mentioned, what it's doing now is it's sorting by the coordinates, right? So each read's alignment is sorted based on its position on the reference genome. And we can show you that in just a second here. So if we do samtools view of this first file, so samtools view will print the file to the screen in SAM format. I don't want to read the whole thing. I'm just going to look at the first 10 lines. There's your first 10 alignments. And one thing you'll see very quickly is they go all kinds of different places across this chromosome, right? This is 781,000 is the first coordinate, 790,000 is the next coordinate, and then 926,000, and then 2,390,000, et cetera, like all over this chromosome, different places. If we do the same thing again, you'll see that this has been sorted. So now I'm doing the exact same command, samtools view, which will take my BAM file and print this SAM file to this screen. And I'm just going to look at the first 10 alignments. And you'll see that it's completely different reads because it's now been sorted. And in fact, each of these read alignments begin at the very first base pair on that chromosome, right? So 1, 1, 1, 1, 1, 1, 1, 1, 1. They all start at the exact beginning of the chromosome, right? So we've sorted our file. Now we got to do something else. We have this other concern, which is that, as I mentioned, PCR duplicates might be really important. What we're going to do is the samtools rmdup command, remove duplicates. And again, let's pretend we don't know how to run this. If we just hit enter, it'll tell us exactly how to run this command. In this case, what you want to do is hit SAM tools rmdupe. And this time you have to give it a sorted BAM file. So you have to give this a sorted BAM file. The reason is if you wanted to compare the mapping coordinates of all reads across the entire file, you'd have to load the entire thing into memory. And that's not a very convenient way to do this. What you'd really like to be able to do is to compare reads that might map nearby to each other very quickly. And if you sort the file, you've already put all the nearby mapping reads nearby to each other. So it's very quick to do it. So input sort.bam. And then what it wants me to tell it is the output BAM file. So we'll say sorted.rmdupe.bam. In other words, I've sorted this file, and now I'm removing the duplicates from the file. If we run that, it's going to take a second. It'll spit out things like this. You don't need to worry about these. It's in there because of a small compatibility issue, but it basically doesn't matter very much for what we're doing. Okay, and we're at the end. So what it's done is for all of the read pairs that are aligned, we've gone through and removed every single, or all but one of each read pair that has the same first and last coordinate. So in this case, it's telling us how much it removed. And in this case, it's only 18,000 read pairs out of 680,000. So it's about 2.5% of the library, which is not bad. It basically means that this is a pretty good library that doesn't have a really high rate of PCR duplications. It is also possible, of course, to get the same first and last coordinate by chance. And so rmdupe should be considered conservative. In some cases, you might know that your library was made without any PCR amplification, and you might not worry about this. And so there are other ways this can work, but the basic workflow is to remove any read that has the same forward and last coordinate. Okay. Okay. The other thing we can do really quickly is we can we can actually identify some of the some quality scores. So if we just run SAMTools view and we say, let's imagine we want to filter this file. Well, SAMTools view can do that. So for example, we might retain only reads that have a mapping quality of 30 or more. So that's this command here. -30 would be that the probability that that read is mapping correctly is 1 in 1,000. So we can be pretty confident with the alignments that we've retained that they were mapped correctly. And if you do that on our sorted rmdupe BAM file, right, following this set of commands they give us up here, what that would output is actually a SAM file, of course. So let's imagine I don't want a SAM file. If I pipe that back into SAMTools view, I can do it that way, sorry. There's another way to do this, which is just tell it my output format should be BAM with -b .sorted.rmdupe.q30.bam. Okay, so Q30 is my filter. If we do that, we'll filter our file only for alignments with mapping quality 30 or more. Okay, let's take a look at what we made. So we have a couple of different BAM files we've made, right? BAM, sorted BAM, sorted rmdupe BAM, and then sorted rmdupe Q30 BAM. And you can actually use the file sizes here to basically figure out what's going on. One thing I think is worth emphasizing is that when you sort a BAM file, it's typically much smaller than the unsorted version. And of course, the reason is because they're nearby in space. And so we can record that information efficiently relative to how you would record them if you didn't have them all sorted. You see that we didn't remove a lot of duplicates. As you would expect, the files are almost the same size, so not a lot changed there. But Q30 removed actually quite a lot of reads. Almost half the reads had a mapping quality of 30 or fewer. And so we know we have many less, but let's just confirm that to ourselves with another function, which is SAMTools flag stat. And what this will do is it'll go through my BAM file and give me sort of a summary of the important stuff. And this is often really useful for just a quick confirmation of the things you probably already know. If we do that, okay, we're done. It tells us all sorts of useful things. It says, okay, here's how many total reads we had. This is how many failed QC, none, because we don't have QC information in our file. How many supplementary alignments are in the file? It's about 700. How many are mapped? All of them, because I've removed unmapped reads, etc. So it's a really useful quick summary of my file. You can also do that on the first file and see that it's not that different. What did I do? Oh, sorry. Flag stat. If we do the same thing, flag stat on the file. we get this out. We just see that there's a heck of a lot more reads in there, and there's a slightly higher rate of singletons actually after we do our removal than before. But one thing you will see is that the properly paired reads, i.e. reads that map pointing at each other in forward reverse orientation, are much more frequent after we removed duplicates and after we filtered on Q30 alignments than before, right? It's 60% here and it's 98% here. Okay, cool. So the last thing I want to show you here is a simple text-based way to evaluate your alignments a little bit. So if we use SAMTools TView, oh, sorry. We want this, SAMTools index. So what index is going to do is it's going to create a table for random lookup from a sorted BAM file. It has to be sorted because the table is based on the coordinate order in the file, but it can basically tell us what position in the file to go to read a given section of the genome or alignments from a given section of the genome. Okay, that's done. You'll see that what that's done is create this file here, this .bai. That's your BAM index file. Now, this is a really simple way to view alignments. You might find that there are other tools you prefer. Most people use something called IGV, but I want to show you something I can just do in the terminal without flipping around too much. So if you use TView and look at this file, you can actually view your alignments, right? So if we scroll along, this is our reference genome starting at coordinate 1. If you scroll along, you can see that we've actually got quite a lot of alignments in most places. Each of these is one of our reads aligned to the file, and it looks pretty good, right? They all basically match. They all call the same sequence. There aren't a whole lot of differences here. Like each column has the same characters in it, right? These are all G's, these are all A's, etc. The only thing to point out here is that in capital letters are reads that are aligned in forward orientation. Lowercase letters are reads aligned in reverse orientation. So they actually are aligned to the reverse complement of the sequence that you see there. Okay, cool. So they're also highlighting positions where there are differences. So this A is highlighted, and it's because you see the C down here that seems to disagree with the A, but every other read is an A, so probably it really is. This is a useful way to just interrogate an alignment really, really quickly in some cases. So I do recommend it. If you have a region that you think is interesting in your genome or something you want to look at just to see if my alignments make sense, you know, the human eye is really good at pattern recognition, and you might be able to pick out something that's wrong very quickly, where it might be really hard to write a script or a program to do that. So I really highly recommend looking at your data as much as you can. Okay. So if we hit Control-C, we'll leave SAMTools TView. That is, I think, everything I intended to say about this. It's a bit of a scattershot, but that's why you have this file as well. So you can go back and see how we did it and see why we did it. But that's the sort of basic set of commands that can get you from short read files to an alignment file to a reasonably processed, sorted, remove duplicates BAM file in BAM format, which again is binary version of a SAM format file. Cool. All right, guys. With that, I'm going to bring us back to just a few kind of ideas about what the future of short read alignment seems to be, or what I think it is. So you get an idea what the big research questions in the field are and have an idea where we're going next. So one of the most appealing things that has come on the market recently is something called a variant graph. And a variant graph, let's see if I have this. Okay, so a variant graph is trying to solve this problem. This problem is called reference bias. And what it basically is if we imagine that you have a reference sequence, well, your reference sequence is just a representation of one individual in your population or species. Your problem is it is actually harder to discover variation that isn't the same as what the reference has. The reason is because when I align my reads to a reference genome, I'm aligning based on similarity, right? And so if there were a region of the genome that was sufficiently different between the reference and my sample, I might never even be able to align reads there at all. A more subtle case is something like this, where let's imagine you have a sample that's heterozygous at this position where the reference has a T, and this individual is heterozygous TG. What you can see is that all of these reads that match the reference align correctly, but there's only one of the reads that doesn't match the reference aligns correctly with the G here. Presumably, there may be many other non-reference reads that we weren't able to align because of this difference, because we're explicitly searching our reference genome based on similarity. So this is a really important problem. And one solution that people have come to is something called a variant graph. And this is something that Benedict Payton in my department and Richard Durbin and others are currently working on. And I've actually done different things, but I think their solution might be better. So I'm doing this stuff now too. But the way this works is, what if rather than having a single reference genome, like a single individual's genome, we instead try to represent all the variation we see? Right? So we go to our population, we sample it, we get an idea what the variation really looks like, and then we encode something. Then we encode something which is a graph-based structure, which has little divergences, these little bubbles, every time there's a difference, right? So in this case, what they're trying to show you in colors are haplotypes within a population, and these nodes that differ based on the variant that's present at a given site. So all the sequences go through the same part of the graph here. There'd only be one section of the graph from TGC, blah, 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 this chunk. But 2 of them go up and have a C at this site, whereas 5 of them go through the T at this site. So we know there's one variant there. And imagine that my reference might have a T. It would be much harder to see that C. But if I encode that and I build an index that knows about that variation, then it's much easier to recover these things. If we then go back down, you see these 2 go through the A. Most go through the A, except for this purple here, which goes through G. Back up, they all match for a while. And then again, there's some divergences in my graph, right? Where these 2 go up and they go to G, these all go through A, they come back together, they go apart again, they come back together, etc. So this graph kind of has divergences where there's variation, it comes back together where there is no variation that we know about. This still has a type of reference bias, that is to say that you have to discover the variation that you want to display in your graph. But I think we can probably do a pretty good job of that and then come back and evaluate other samples that we're not as sure about and do much better. Okay. An example of this that might be interesting to you was that VG, variant graph, was actually used for an ancient genome as well. And the reason that this works really well for ancient genomes is, well, for one thing, they may have lots of variation that doesn't match our modern reference. But for another, they also tend to have lots and lots of errors and indels that makes alignment harder. So if you at least accommodate the known real differences in the genomes, you can do much better. So this case is actually a large indel that was known previously. So this is a deleted section of sequence that you see in these reads here. When you align those same ancient DNA reads with BWA, you don't get that at all. They don't get aligned to the reference. But with VG, variant graph, you do. You actually see that that indel is represented very cleanly, and it's because we knew about that indel in the first place. So it was possible to align our reads around that sequence without having any major issues. Okay, these are just some results from their first paper on this. We see something pretty similar in our own data, but basically what they're doing is they're showing you something like a ROC curve. It's not an actual ROC curve because it's both sensitivity and specificity. And so what you're doing is I'm misspeaking, sorry. Reads that don't align at all aren't in here. So what you get are these lines that don't extend all the way to the bottom and all the way to the top like a traditional ROC curve. What they're doing is they're stratifying their true positive and false positive rate for the reads that did align successfully based on the quality score, right? So Q60 tends to be the highest quality score. That would be probability error 1 in a million, all the way down to Q0 where we're pretty sure it's an error. And of course, as you would expect, your false positive rate goes up as you become less stringent with your filtering, but the true positive rate must also go up at least a little bit because you align all your reads. So you have to get a few of them right and it goes up as well. But the important point here is that if we look at the Bois mem, paired end, and single end functions that are over here, they sort of always do worse. They live in down here in this space of this plot. And importantly, your false positive rate's log scaled. So we really want to be as far to the left as possible. Your true positive rate is not log scaled, so it's a little less concerning. Almost everything is finding the same true positives, but you can get a lot more false positives. But VG is these things up here. So what I'm basically trying to say is, yes, quantitatively, variant graphs do a lot better than single linear reference genomes. And I think it's it's still up in the air exactly how these should be done. There's lots of different algorithms that are being developed, but I definitely think it's going to be the future of how we do variant discovery and short read alignment to a reference genome, particularly for species like humans, where we really know where the vast majority of the common variation is in the human genome. So it isn't about discovery, it's about accurate genotyping. And so what we'd like is to be able to find all those spots and accurately genotype them. Okay, so another thing I think is really exciting, unfortunately, we're not gonna have time to get to my new research today. I'll just take questions at the end. You could ask me about my research anytime, though I love talking about that, as we all do. But what I'm going to talk about a little bit is some new molecular biology techniques that have also enabled some pretty cool new kinds of data that are really interesting for metagenomic applications, but also for healthcare related applications as well. And so what these are is something called a unique molecular identifier. What that is a little section of DNA, an oligonucleotide that we synthesize, and we synthesize it to be random. So in other words, these may be 16 base pairs long, but it's 16 at random A, C, G's, and T's. The reason you can do this is when I ligate that to a molecule, if I then do PCR on this sample a bunch of times over, there's a pretty good chance that I will sequence the same molecule a few times. But because I have a unique molecular identifier, I don't need to use the alignment to the reference genome to figure out that it's the same molecule. I can actually just read this barcode that I've added, this 16 base pair barcode, and I know for a fact that those sequences must have all descended from the same progenitor molecule. Okay, you might say, why do I care? Well, let me explain. This is a specific version of this that's even cooler than typical UMIs. It's called duplex sequencing. The way duplex sequencing works is you make an adapter that looks a lot like a standard Illumina adapter sequence. And it actually has the standard read primer sites right out here. So your read primers would sit here, but it also has, before you get into the actual read sequence, a randomized duplex tag, right? And so when it says duplex, what that means is that both the forward and reverse strand have that same sequence. Well, obviously the reverse is the reverse complement, but they are the exact same tag sequence. And so what you do is you ligate these adapters with TA ligation, so sort of the standard way we do molecular ligations, and you ligate them to both ends of a DNA molecule. So you have an alpha and a beta, each of which has this randomized duplex tag. And so what you can do then is, for one molecule, you can amplify this. And actually, in this case, what we're doing is we're intentionally creating PCR duplicates. So we limit the amount of material we put into our PCR reaction, and we do an absolute ton of PCR until we have grown these families, right? Now, why is this great? Well, the reason this is so cool is if I read the bottom strand, you'll start with primer 1. You'll read this way. It'll go beta, sequence, then alpha. If I go from the other strand, you'll go alpha, the DNA sequence, and then beta. These, of course, should be the reverse complement of each other. Now, if I collect up all of my reads at the end that have the same alpha tag and the same beta tag, what we can do is make a consensus out of them. So it's called a single strand consensus when you just make this first one as alpha and beta. And you do the same thing in reverse with the beta then the alpha. At the end, you'll see, okay, alpha and beta, there's these other little differences. These are either sequencing errors or amplification errors, but all of them have that green variant there. Therefore, we're really confident that that green variant is real. Even more confident than we would be with traditional sequencing because we've seen it 5 times over. We do the same thing with the other strand, right? So the molecules that descend from this bottom strand. And again, we see that same green variant in all of them. So we're really, really sure about that. But we also see this brown variant. What that probably is an error that was incorporated in the very first round of PCR. That's why you only see it on one strand and not on the other strand. So when you make your single strand consensus, you'll say, okay, this is the green one. This has green and brown. But when I make my duplex consensus, that is, I take the consensus of these consensuses, we'll say, oh, we only trust that green mutation. So why does this matter so much? That's the same thing. Again, don't worry. The reason this matters is if you wanted to know about really, really rare variation in a sample, like somatic variation that you see in a tissue from a normal adult human, you couldn't do it because the error rate would be so high. And in fact, this is a comparison of the two using standard Illumina sequencing, where what they're doing is they're showing you the percent variation at a given site across a relatively short, it's about 300 base pairs or 250 base pair section of a gene. And this is a gene that we know confers cancer susceptibility. And there's a known driver mutation in this gene. And what you can see is that if we just sequence our tumor sample, nothing really stands out. There's just some rate of variation across this whole thing. And so it's very hard to know what to make of this. But when you do duplex sequencing, because you can vote out all those errors, because you aggregate all these molecules that you know descend from the same original molecule, and get rid of all the error, what you're left with is just this one site at a modest frequency, a little less than 0.2%. But we're pretty darn sure it's real because we saw it many, many times over in many different duplex consensus molecules. Okay, and so what that gives you is extremely accurate sequencing, even to depths of sequencing of many thousands X. Even if you only see a few reads that confirm this alternate allele, we can be pretty darn sure that alternate allele really does exist. So that's awesome. Okay. Okay, so what we're going to do now, I don't know, how am I doing for time? Let's see. We got a few minutes. Okay, the last thing I'll talk about is one of the newer things that people are doing more commonly is called haplotype phasing. The way phasing works is that when you sequence a genome, you have usually diploid genotypes, right? So this individual has two alternate alleles relative to the reference here. They're 1, 1, but they're heterozygous here, 0, 1. They're homozygous here, heterozygous here, etc., right? So each of these 0, 1s is a case where it's a heterozygous variant relative to the reference. What you can do is called read-backed phasing, where we know a read descends from a single DNA molecule. It can't possibly come from both chromosomes in a diploid individual. It must come from one. And so what we do is we align our reads and we say, well, which variants does that read confirm? In this case, you see that this one confirms 0, 1, 1. So we can be reasonably sure that those alleles are all on the same chromosome. This one only links 0 and 0 for some reason. This links 0, 1, 1, etc. But if we use all of those and we come down here, you can see that we can actually tease apart these two. So instead of having these diploid genotypes where 0 and 1 could be on either chromosome, we figure out that they are on either the maternal or paternal chromosome by teasing them apart. Okay. I think that's probably all I'm going to have time for today if we're going to answer some questions. So thank you all for coming. And yeah, we're almost out of time. Yep, that's going to be it. Does anyone have questions for me? There we go.
Well, thank you very much, Russell,
Sure.
for 3 brilliant lectures.
Yeah, some were better than others, but that's all right. Thank you. Figure it out.
Yes, and well, if people do not write any and ask any questions now, so maybe if they have them later, they then can send you an email, right?
Yes, yeah, send me an email, that's fine. You can find me on my lab's website pretty easily. And I'd be happy to give you more information about any of this stuff. Like I said, the Burrows-Wheeler Transform is awesome, but also a little bit, you know, I explained the very short version of it. There's a lot more there and other stuff, of course, as well. And so I think I'd be happy to give you more information or point you in the right direction. All right, guys. Well, thank you.
Thank you again, Russ. It was like very nice to have you teaching us.
Cool.
And yeah, the next 3 lectures will be by Torfinn Corneliusson from University of Copenhagen.
Awesome.
All right.
Everybody.
Thank you.